My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. We have a good one today. I am sitting here with Dr. Laurie Vitt, who is an Emeritus Curator from Sam Noble Museum at the University of Oklahoma. And Laurie fits into this category. You know, we've done interviewed a number of herpetologists, a number of snake ecologists who are kind of, people who have just had really incredible, productive careers, really added to our knowledge of these animals. And, I really wanted to chronicle, interview them about their career and just kind of get their perspective, on how they ended up where they are today. And Laurie definitely fits into that. He's someone that I haven't met before, but I've, certainly read and used his books extensively and read many of his publications. A name maybe that I've looked up to over the years. So pretty excited to chat with you today, Laurie. How are you doing?
Pretty good. I'm a little bit allergied out from mesquite trees, but other than that, I live in southern Arizona now.
Oh, okay. Whereabouts in southern Arizona?
A little town called Tubac, which is about 30 miles north of Nogales.
Oh, great. Okay. Yeah, I was just down in that part of the world not too long ago. I gave a seminar at the university there in Tucson, and then one up at the Phoenix Zoo, and spent some time out in the Coronado. And yeah, so beautiful part of the world you live in. But so I like to start off with all my guests and kind of get a feel for how they ended up getting into snakes, herpetology in general. Again, for most of society, it's actually a pretty eccentric or outside the box type of thing. So I'm just curious how that happened. Is this something, were you always into these animals when you were a child, or is it something that kind of developed later on in life?
I started off when I was about 4 years old. I caught a garter snake in our yard, and I thought that was the coolest thing I'd ever seen. And from then on, I was out looking for animals, turning rocks over, bringing things home. And I guess early on, I thought, I want to be one of these guys that works in zoos and so on. That was my kind of vision of what, animal biology was at the time. And then, as I got older, I got more and more interested in research.
Gotcha. So you found that first animal under the rock there. So what part of the country did you grow up in?
Billings, Montana, of all places. And
oh, really?
Yeah, and Billings, of course, has a low diversity of reptiles and amphibians. But by the time I was about 9 years old, I think I had collected or found every species of reptile and amphibian that occurred there, most of the small mammals, scorpions, centipedes, anything you can imagine. I brought porcupine home once.
That's great. Yeah, I've spent a lot of time in Montana. I was actually just in Montana a week ago. Billings, that's kind of an interesting area as well. People think of it as low diversity, but, I worked in the Northern Rockies a lot, and I always thought, what the Northern Rockies lack in diversity, they make up for in abundance. And just like, abundances, some of the things you can find, like rattlesnake abundances, for example, and densities can be really, really high, which is fascinating to me.
Right.
So you said that you found that garter snake growing up. So that would, over in Billings, would that be Western terrestrial, like elegans, or would that be
No, it was Sertalis.
Sertalis? Yeah, because elegans is like the interior, intermountain west.
Yeah, elegans actually occurs in real high densities along the Yellowstone River. Where they dredged
Okay.
stuff out and piled it along the river. You can walk along there, and on a sunny day, all of those piles of debris will have elegans in them. And
Oh, okay.
that's the only place I ever got elegans when I was a kid.
Oh, okay. Interesting. Well, one thing I've done a number of times, and I don't want to say where, but there is a predator in, they always talk about Yellowstone having its full suite of predators, but one they never mention is that there are actually rattlesnakes in Yellowstone National Park. And again, I'm not going to mention where they are, but I've been visiting a lot of those hibernacula and gestation sites for a number of years. And I've always appreciated Montana. And the other thing I'd say about Montana before we leave it, you're in Billings, so you're still pretty far west, but if I remember correctly, that southeast corner of Montana, you actually get some kind of interesting things popping up into that corner, things people wouldn't think of in Montana. Is that right? I'm thinking like some turtles, maybe like snapping turtles, or am I wrong on that? But anyway, some interesting.
Yeah, I think you're correct. And there are a couple things in Billings that kind of stop at Billings. Western hognose snakes, for example. There's a milk snake there, and when I was a kid, I found quite a few of those, and then did some work there in I think early 2000s for BLM, and found the crew that I had. We found I think 7 during the summer that we were there, and they were always difficult to find. You turn a million rocks, and then you find a milk snake.
Mm-hmm. Yeah. Huh. Sounds familiar. But anyway, so you're growing up in Montana in the frigid north, if you will, and but you're fascinated with some of these animals that a lot of people really could care less about. But you're developing this interest, and it sounds like at an early age you even realized that this was potentially a career, but you were thinking about it kind of through the lens of maybe working at a zoo or something like that. So how did you pursue, that idea of a career with them? Did you end up going to school right away, or did you end up working with them somehow? How did that all come together?
Well, I always collected reptiles and amphibians. And when I was a teenager, I moved around. My parents moved around. They were teachers. And we moved to Tracy, California, and then to Sacramento. And in Sacramento, I met a guy who ran a pet store, and he imported all kinds of things. And that was back when you could import anything. And so I ended up with a bedroom full of cages, and I had a lot of really dangerous snakes, and I think I didn't realize how dangerous they were. I had a gaboon viper, I had puff adders, African puff adders, I had European vipers, and I was just really fascinated with all of those sorts of things. The first time I saw a reticulate python, it just blew me away, all the colors on those things. So I had a reticulate python that started off at about 4 feet, and the next thing you know, it's about 12 feet. And I finally had to give it back because it was just too big to handle. But I went through a phase of just having a big collection. And then when I went to college, I majored in biology, and I continued to go out and find things. And I was at Western Washington in Bellingham, and I would go over the mountains to rattlesnake dens. I had a friend who worked at the Woodland Park Zoo, Ernie Wagner, and he was one of the people who first started breeding the banded king snakes, the red, white, and black king snakes. And so he and I went out to a lot of places, including southern Idaho, and, found really neat things in the field. So I kept doing that the whole time that I was an undergrad. And then I worked for a year in an electron microscopy lab in Seattle. And my job was to basically prepare the scopes in the morning for the doctors that were working. And I brought a rattlesnake in, and we did a perfusion of the rattlesnake with glutaraldehyde and preserved all the tissues in the snake. And then I started looking at all the different tissues in the snake and with an electron microscope. And so I learned a lot of cell biology during that year. Then I went back to Western for a master's, and
Western Washington? Or?
Yeah, Western Washington and Bellingham. And that's where I got interested in lizards. I wanted to work on snakes, and I wanted to work on rattlesnakes, which I think most snakers, do when they're young. And my advisor convinced me to switch to alligator lizards because they were common around Bellingham. And so I started working on those, and they were live bearing. That got me interested in life histories. And so, the next thing I know, I started applying to graduate school, after I got a master's, I applied to PhD programs, and I had met Chuck Lowe at University of Arizona a couple of years earlier because one of his students was the ecologist at Western. And Chuck was, I don't know if you've ever met him, but he was an impressive guy. He was really tall, 6'6", I believe. He was an ex-UCLA basketball player. And in a lot of ways, he was crazy, but he was always brilliant. He was also brilliant. And I wanted to work with him in Arizona, and I applied to U of A. He told me to send the application directly to him, which I did. And at the time, I had real long hair, and that was in a time when, if you had long hair, you were considered a hippie. And he never responded. And so I applied to ASU and got in ASU, and then started working at ASU. And I got really interested in life histories for a while, worked on lizard life histories as a graduate student. Met a couple other really well-known biologists. One was Ernst Mayr, who was an evolutionary biologist, and he spent a year at Arizona State. And then Donald Tinkle, who was a really famous herpetologist from University of Michigan, and he spent a year at Arizona State, and that got me more and more interested in evolution. And I've been interested in evolution at Western as well. But it was really at ASU and the interactions with Don Tinkle, I think, that kind of cemented that. And from that point on in my career, I worked on a lot of different things, but sort of behind everything was an evolutionary perspective, asking questions about why and how kinds of questions rather than just what kinds of questions.
Yeah, I want to get into that, but I want to back up a little bit first. I want to go back to your Western days. And I'll tell you, when, in researching for this interview to do this episode, when I Google you, there's two things that pop up. Herpetology related things and music related things. And we don't need to get into your music, but there was a picture, I think it was from a band that you were in. It was a YouTube video from a band you were in. I think it was when you were in Bellingham, and you were on stage with these other band members, and you had a really large snake, and you were kind of holding it over the band. What was that snake? I couldn't idea it.
Yeah, it was a boa constrictor, and
that's what I thought.
Yeah, it traveled with us, and the band I was in actually did pretty well on the West Coast for about 4 years. We toured up and down the West Coast while I was an undergrad.
That's great. Okay. So let's go. So you did your master's at Bellingham as well.
Right.
Correct?
Yeah.
On the alligator lizards. So that's when you started working on lizards. So talk to us, what did you do? What was your thesis research with the alligator lizards?
With alligator lizards, I worked mostly on reproductive biology. And, Western at the time really wasn't a research university at all. It was actually called, I think, Western College of Education or something like that. The name was different than it is now, and I had some really good professors there. They were mostly teaching professors. And when I entered the master's program, my grade point as an undergrad was so bad that I had to beg my way into the master's program. And I was basically told immediately that I could not get any grades below a B. and of course, most of the professors knew that I played in a band, and I was traveling up and down the coast every weekend while I was an undergrad, and that was part of the reason for my poor grades. And so in a master's program, we had to take qualifying exams, and everybody was scared to death of those, including me. And so I studied my brains out, and I got the best score they had ever seen on the exam, and the result of that was that all of a sudden I really got the attention of a lot of professors, and they really helped me out a lot. And so Western turned out to be a really good place for me at that point in my career, because I was at a stage where I was trying to decide whether I should take a music direction or I should take an academic direction. And my older brother was a professional musician, and he did real well his entire career. So I had those kinds of connections through him. He was a studio musician in California, and I had connections from the band that I was in. And so it was a difficult choice to think, should I move on and pursue biology, not knowing what kind of a career it could be. And so that was probably the most difficult decision when I was both an undergrad and a master's student. But music supported my education habit.
Yeah, interesting. Well, alligator lizards have always been interesting to me. I know very little about them, but I remember Chuck Peterson, who's been on the podcast, in the past. But I remember him using them as an example of an interesting animal, talking about species distributions, where you need to think about different life stages and complexity, and that based on their thermal biology, he thought that the adults could actually live, say further north, but it's the idea that the young could not, survive at colder latitudes or elevations is what held them back. Is that accurate? I've always kind of told that to people.
Yeah, that's close. What happens is that the species that occurs the farthest north, which is what I worked on, is live bearing, and so they don't deposit eggs, and it's the eggs that would never hatch in those cold environments. And the females actually come out even in the winter and bask on crevices. They spend the winter deep in crevices, and they come out if it's sunny, and that helps develop the young. And if you go south, that species goes into Northern California, and it's replaced by a larger one that lays eggs. And so all of the other species in North America lay eggs, and they live in warmer environments. And here in Arizona, for example, we have a species it's the Arizona alligator lizard, and it's a really pretty alligator lizard, but it occurs in the mountains, which are warm in Arizona, and it lays eggs.
Okay, interesting. Well, back to your timeline. So you finished the master's, and you evaded the allure of a musician's life on the road and ended up studying biology, going on for your PhD in Arizona. And so what was it that you studied in Arizona for your PhD?
Donald Tinkle was interested in a thing called reproductive effort, which had to do with, how much and why do animals invest a certain amount of energy into reproduction as opposed to other things, growth, maintenance, and so on. And so I was interested in that. And he at the time thought that one of the answers would be in looking at the calories in eggs, how much energy is in eggs. And he thought that there would be differences in the, per unit weight density of calories that would give some clues as to the evolution of reproductive effort. And so I started looking at that, and I collected a lot of lizards. I collected a lot of eggs. I spent a lot of time in the lab looking at calories in eggs using a calorimeter. You basically burn a sample of the egg and determine the amount of calories. And as it turned out, he was totally wrong in that the calories in eggs of different species of lizards were all the same. In other words, caloric density didn't vary. And of course, what did vary is the number of eggs, the size of eggs. Now at this point in time, we know a lot more about it. At the time, we didn't have a lot of the tools that we have now. For example, molecular phylogenetics wasn't even around back then. And so a lot of the questions that we would like to answer or would have liked to answer back then, we couldn't do because we didn't have the tools. So that's mostly what my PhD dissertation was. And I kind of lost interest in life histories after that, although I've come back to that with some of my colleagues, my Brazilian colleagues, and we've done some different kinds of analyses looking at the evolution of life histories using molecular phylogenetics. And I have some collaborators that do these phylogenies and using a lot of different genes. The phylogenies have become pretty darn good, fairly solid. So,
yeah, I do want to get into your tropical work as well, but we'll do that later. But it's interesting. There's kind of this progression through your life and career with snakes and reptiles, like, cold places just continually getting closer to like warmer, more biodiverse places. So that's kind of interesting.
Hey everybody, I just wanted to take a quick break and tell you that the Oriane Society is dedicated to protecting some of North America's most at-risk species and their habitats, like eastern indigo snakes in the longleaf savannas, eastern hellbenders in the Appalachian highlands, and wood turtles in the great northern forests. Healthy ecosystems depend on these incredible animals, but these landscapes and species need our help to survive. If you care about reptiles, amphibians, and the wild places they call home, visit www.oriane.org to learn more about our work and how you can get involved.
So you're in Arizona, and first of all, as I mentioned, like that, you must be going crazy. I mean, you're doing these studies, you're talking about lizards, but you must be just really overwhelmed by the diversity, the snake diversity, the lizard diversity, as compared to like Montana and Washington. And so you finish up your PhD in the life history work, and then what happens between then and ending up at the museum there in Oklahoma? How did that trajectory?
Yeah, I did a series of postdocs, and the first one was in Brazil. I spent a year in the state of Pernambuco in the northeast part of Brazil, and it's a semi-arid area. At the time, it was the most poverty-stricken area in the New World, and there were 3 other Americans working there. They were working on mammals, and they were students of a professor named Michael Maras, and I actually had met Michael Maras when he gave a seminar at Arizona State when I was a student, but he wasn't in the field. And so I showed up in this remote town. These 3 other Americans were there. I didn't speak a word of Portuguese, and I was just embedded in this little town. And it turned out that it had a pretty good diversity of lizards and snakes. And so I started working on all of the lizard species that occurred there at once. Since I was there 24 hours a day for a year, I had a lot of time to work on them. And what I found, a lot of the theory on life histories with lizards had to do with the impact of local environments. In other words, local environments might determine what the life history is, kind of like with alligator lizards along the West Coast. And so I sort of went in thinking, well, all of these lizards are going to be doing pretty much the same thing. Well, it turned out that I was totally wrong. And in fact, I was able to show that every species was different. Their life histories were quite a bit different. And in addition, every known life history in lizards was present at that one locality, plus one that was unknown. I discovered a skink that ovulated eggs that were about the size of a pinhead, and they developed completely inside the body of the lizard like a mammal, very much like mammals. And when I got back to the United States, I started a postdoc with Don Tinkle at Michigan, and I told him about that. And I had this crate with all of my samples in it that had been shipped. And it was in the basement of the Museum of Zoology at Michigan. I told him about this skink. And I said, this thing is viviparous like a mammal. And his comment was, no, it wasn't. And I said, well, yes, it was. And I said, I'll show you. And so I went down in the basement and I dug through my samples, and I brought up a bunch of samples of the oviducts of this skink that I had brought back. And, when they first ovulated, it was just a speck in the oviduct. And every month they got bigger, and it turned out to have a gestation time of 9 months, and a little over 9 months. And, and then it produced live offspring. And so I ended up getting together with a physiologist and a morphologist, and we described that. I gave them the samples to look at, and we published a paper in PNAS. We sent it to Science, and it got really good reviews, and the editor rejected it. And I called the editor up and asked why it was rejected, and she said, it must have gotten bad reviews. And I said, let me read you the reviews. And I did. And she said, well, you have to understand that right now developmental biology is hot stuff, and that's the kind of stuff we're doing. And she didn't think it was the kind of stuff they were publishing. So we put it in the Proceedings of the National Academy of Sciences. But that was really a neat accidental finding. And, one of the things that I've discovered, and you'd probably get the same comment from Harry Green, is that, the animals tell us what's going on. And, we go out as naturalists and we study these things. And if we keep an open mind, we see things that we would have never imagined if we, had a closed mind. If we went in thinking, well, this is the way it works, it's got to work this way. But, so I've always seen myself as more of a naturalist than anything else. But
yeah, I think even like the titles to Harry's Snake Book, for example, probably would show just what you're talking about there, kind of discovering these mysteries in nature and letting nature tell you what is out there. A quick question for you. So I give a lot of presentations to, whatever, scout groups and big presentations to, community groups outside of the scientific community. And, most people talk about live bearing, and I, present a lot on oviparous versus ovoviviparous animals. And I talk about viviparous, but which snakes, do you know which species of snakes are more viviparous than ovoviviparous? Kind of like the skink you're describing?
I don't, but I know there are some, and there are none that ovulate really small ova. There are some that ovulate, ova that are part that have a placenta associated with them, but the eggs also have a lot of yolk to start with. And so I don't know of any snakes that are anything like these skinks. And we now know that a lot of the South American skinks are actually like this, and some of the African ones are as well. Once we published a paper on this, people went back and started looking at these things and realizing that there were quite a few species that did it.
Yeah, it's amazing. So, let's. I want to come back to the tropics because I think you've continued working there. But I want to figure out, so you're doing this postdoc, you're in Brazil, and then you end up in Michigan. Did you end up going from there to Oklahoma, or were there more steps in between?
Yeah, I went from there to Athens, Georgia on a different postdoc. And in the meantime, I was writing up a lot of my tropical stuff. And then I got offered a position at Savannah River Ecology Lab, which is part of the University of Georgia, as an ecologist. And at the Savannah River Ecology Lab, I took over the program that my wife had started, which was this big amphibian program. And she had taken a job at Emory University as an associate dean. And so I took over her position and ran that program for a year. And that was a massive program. We published a paper on it in Science that kind of was a culmination, I think, of 12 years worth of work. And then I interviewed for a job at UCLA that I was convinced that they were going to hire Mike Ryan. And I was even told that by one of the people when I interviewed. And on the last day of my interview, they sat me down and they said, "We're going to offer you this job." And Mike Ryan just happened to call the same day and say that he had taken a job at University of Texas. And so then I went to UCLA. And
okay.
Yeah, and I spent 8 years at UCLA. And at the time that I was hired there, there were several people, George Bartholomew, a physiologist, and a few other people, and they had a policy that they did not consider grant funding in promotions. They considered research only, and research and teaching. And so I did really well there because I was publishing a lot of work. I continued to work some in Brazil. I started working in the Amazon. So I moved up through the ranks pretty fast. But remember the Mike Mars person from Brazil, whose students I was with on my first trip in Brazil. Well, Mike called me one day and said, "Hey, we're building a new museum, and we're trying to build a new museum in Oklahoma, and we're going to have a herpetology position. Do you want it?" And I said, the problem is that I'm building my house right now in Topanga Canyon, which is true. I was building it myself and building it out of pocket, and I said, it's not quite finished. And I said, I just can't walk away from it right now. And so they didn't hire anybody. And a year later, he called again. And he said, we've got this position, and if you want it, it's yours. And he said, and I can work on a second position for your wife. And so that's why we left UCLA. I was there 8 years. And
Was
I like.
the UCLA position also at a museum, or was it
No, it was a
primarily
faculty
teaching faculty?
position. Yeah, but
Yeah.
the way it worked there, we could arrange teaching so that we could be in the field. And so I usually did all my teaching in one semester, and then I'd spend, 2 or 3 months in Brazil. And at the time, Brazil was funding a lot of the work that I did, so I didn't write grants or anything. I just wrote papers based on research that I did. And my wife went with me. She worked on frogs. And and when we left, I was a little bit reticent to go to Oklahoma because one of the first things they said was, well, you're going to have to get grants. And, I was kind of tuned into not having to do that. And, when we got there, we wrote a grant. The first one got turned down. And then we wrote another one and learned a lot from, writing the first one. And we were totally shocked when we got it. And from there on, we got really good at writing grants. And so we just got funded out, one time after another from National Science Foundation. And we paid salaries for Brazilians that worked with us in the field. We bought vehicles in Brazil. We bought computers for some of the people. We bought super cool freezers for some of the people. And so we did a lot with curators of various museums in Brazil where we were working. So that worked out pretty well. And
so when you came to Sam Noble, then when you made that transition, was it an existing collection from the university, or were you starting from scratch, building up the collection there?
There was an existing collection, and the collections for the entire museum were in 12 different buildings, and there was no single building at the time. But the director, Mike Mars, convinced us that he was going to be able to get funding to build a new museum. And sure enough, over the next couple of years, he was able to do that. And so we were able to design our own labs, our offices. The building was half research collections and half public. And the museum there it's spectacular. I mean, it'll blow you away. If you ever go to Norman, Oklahoma, you should visit the Sam Noble Museum, and you should ask for a back of the house tour so that you can see the research part of it. But it looks like any modern museum, and it took years to put it all together, but we all worked on different things. We worked on exhibits and so on. And at the same time, we kept our research going. And we also had to teach in the zoology department. But I taught one course a year. My wife taught one course a year. And so the teaching part was relatively minimal. And we were both workaholics. We'd go home and work till 9 or 10 at night and, get up early in the morning. We'd be the first people in the museum. That's just the way we were for 21 years.
Yeah. So let's dive in a little deeper into Brazil. It sounds like that's starting in that postdoc that you had that first experience in Northeast, but then worked in the Amazon and other places. So how would you kind of characterize your work in the tropics down in Brazil over time? Like, what was kind of the focus? Was evolution kind of the backbone, or the life history components? What did you do down there? How would you characterize it?
Well, I'm a real data collector. And so the funding that we had for all of the Amazon work was kind of under a biodiversity program. And so what we were supposed to be doing, and we did, is we'd go into really remote areas, build a camp in the forest, live in an open air camp for a couple months, and then run trap systems for reptiles and amphibians so that we could actually determine the diversity at a particular area. And we got really good at that. And we also collected a lot of data. And so for every individual lizard that I would collect, for example, that would go into a museum collection, I would open the after killing and preserving the lizard, I would open the lizard, identify everything in the stomachs, get reproductive data from males or females, and I had gotten a really good entomology background at Western when I was an undergrad. And so it was really easy for me to identify everything in the stomachs. And turns out my wife also had a really good entomology background. And so she was doing the same with frogs. And so we'd make these large collections in these remote areas and collect a lot of data from all of those as well. And we did other things also. We took temperatures of animals that we collected. We, in some areas, we would use these electronic devices that you could, put down inside of a big termite nest where geckos were living and find out what the temperatures were in those. And so, we were always kind of looking for, neat little studies we could do while we were doing the biodiversity studies. And the Amazon was pretty interesting. We spent a lot of time hiking trails at night, and we'd find a lot of things at night that were active. Most of the snakes were active at night. We found things like Emerald Tree Boas, which just blew me away. The first one we found was a juvenile, and it was bright orange. And, they're just such fantastic snakes. And, we
see
found
any Bushmasters down there or no?
Yeah, I came really close to stepping on one night. And we were walking on a trail right outside of the camp. And I came around a corner in the trail, and we had headlights on. And the snake was coiled up right in the middle of the path, and I almost stepped on it. I backed up so fast, I knocked everybody over that was with me. And so then we were trying to figure out how to get the snake. And so I got this log. I picked up this log, and I thought, well, maybe I can pin it with this. And, as soon as I touched the snake, it was gone. And it had gone way under this underbrush. And we wanted to collect it because it was, a specimen from this biodiversity study. And so I looked way back up under there, and I could see the snake, and it was just sitting there. And so I had my grad student go back and get my BB gun. And I used to use BB guns in the field. And I actually hit that thing right in the head with a BB and killed it just like that. And so we got that as a specimen. And we've gotten them in other areas, and I don't think I've ever seen one under about 7 feet long. They're the ones that we found were always just huge snakes, and they're a lot like rattlesnakes in terms of handling them. They're pretty darn easy to handle. The fertilances, all the different species of fertilances, it's a different story. Those things, you get anywhere near them and their head turns and they look right at you, and they're just always ready to strike. And so you have to be really careful with those.
Yeah. No, I agree. I've handled both of those animals, and I've spent a lot of time looking for both over the years. But so that's great. So you're down there doing all these collections. So did you? So you're kind of documenting biodiversity, and I'm assuming calculating different biodiversity metrics. But then as you bring these animals back, I'm assuming you bring some of them to Oklahoma and place some of them in local museums, however that distributes.
Right.
But are you continuing to do research on these collections over the years after you collect them, or how does that work?
Hey everybody, I just wanted to take a break and tell you guys that snakes are one of the most persecuted groups of animals in the world, but we are dedicated to changing that. The Orianne Society works every day to ensure there's a future for these amazing animals. If you care about snakes, wildlife or wild places, and you like what you've been hearing on the Snake Talk podcast, support us today by donating on our website at www.orian.org.
I don't do much on them, but a lot of other people use the specimens. And so, if somebody's working on a particular group, say a species of lizards, they'll borrow all of the specimens that we have of that particular species and look at them. And a lot of times they discover new species that way, things that are actually morphologically different. And the same is true with frogs. We brought a lot of frogs back. And with frogs, one of the things that we collected are frog calls. And so for most of the species of frogs that we collected, we also have their calls. And their calls are used by a lot of different people as well. And they can be indicative of, a different species if the calls are different than things look alike. And in amphibians, there are a lot of frogs that look really alike, and they're actually totally different. They're isolated reproductively and occur in different areas and so on. So the specimens get used, they can get used basically forever. They're borrowed for a lot of different things. One of the things that we did later with the specimens is, since I had all of the stomach contents, we started looking at parasites. And so we published a number of papers describing parasites in lizards, including new species of tapeworms and roundworms and those sorts of things. And so, all parts of the animals get used. One of the other things that we collected on the biodiversity studies is tissue samples. And so we'd take a little piece of muscle or a piece of liver and put it in liquid nitrogen. We had these big liquid nitrogen tanks that we took in the field. And those tissues have been used continually. They're still being used today, because they take a tiny sample and then sequence the genes and use the genes in different kinds of studies. And so the tissues have turned out to be really valuable. So we try to, our philosophy on collecting animals was always, if we're going to collect an animal, we want to get the maximum amount of data we can out of the animal. And so we've always tried to do that. And, I've given talks a couple of times where people get up and they say, don't you feel really bad about, killing these animals? And of course I do. But almost every area that we've ever worked in no longer exists. And we've had collections from areas that have been converted from tropical forest into basically cattle land. Everything was cut, burned, and then converted to grassland. So the places that we've made collections from, for the most part, don't exist anymore. You can't go back there. Our collections end up being the only collections from those areas. So the collections end up being really useful, in the long term, even though we may not be the people using them. Other people use them. Often we collaborate with people, but, sometimes we don't even collaborate. They just borrow the tissues and do things with them.
Yeah. Huh. So is Brazil the primary place you've worked in the tropics, or have you traveled to other parts of Latin America or other continents?
We've worked in Ecuador, Nicaragua. We worked in Nicaragua right after the war. That was interesting. We've done a little bit of work in Costa Rica. Not a lot of work in Costa Rica, but I went down with a friend of ours, Bill Cooper, and he's a behavioral biologist. And we went down and did a bunch of behavioral studies with both lizards and frogs in about a 2 or 3 week period. He's a really good experimental biologist. And so he could say, here's what we have to do and how many times we have to do it. So we did a bunch of different experiments on things like predator escape and so on. And Bill and I had actually worked in South Carolina at the Savannah River plant, we got really interested in five-line skinks. There were 3 species that occurred at the Savannah River plant. And I think we did almost anything you could think of to do with skinks. We did tail regeneration studies because the juveniles have bright blue tails. And when they reach sexual maturity, they lose the blue color. So we did a lot of experiments with that. One of the things we discovered that was neat is that with juveniles, if you remove the tails from one group and compare them to a group that has tails, compare their growth rates when you give them both groups unlimited food, you would think that the ones that lost their tails would grow more slowly because they have to regenerate a tail. And they have to invest energy in regenerating a tail. The experiments that we did showed that it was the opposite, that the ones that were missing a tail actually grew faster. They regenerated tails and their bodies actually grew a little bit faster. And so they're apparently losing their tail causes some sort of physiological response, which is kind of neat. So Bill and I did a lot of stuff like that in South Carolina. And we also did a lot of stuff on parental care because the skinks actually brood their eggs. And you can bring them in the lab and set them up and they'll do everything they do in the field right in the lab if you set them up right. And so then we'd hatch all these eggs and have all these juvenile lizards that we could do experiments with. And we discovered that they did really well in captivity on termites. So, we'd put a petri dish full of termites in a cage and all the juveniles would all be around the petri dish like dogs eating the termites. So, they turned out to be really good models for doing a lot of different kinds of things. And then, Bill worked with us, later on behavioral studies when we went to Costa Rica. And he and I went to Spain one time and did some work in Spain on behavior of lizards. And so, I've had a number of collaborations, long-term collaborations with different people. And now I mostly collaborate with Brazilians.
Gotcha. Oh, okay. Well, so you've you've spent an entire career going on these, great adventures, it sounds like, in the tropics and living in jungles with, and kind of off the grid type places. You must have some great stories of just interesting close calls. You already mentioned the Bushmaster close call, but are there any kind of interesting situations that you ever found yourself in as part of kind of the bigger adventure?
Yeah, you want a good snake story? Are you ready for that?
I do, but not quite yet. Save the snake story.
Yeah, okay. We've had a lot of adventures. We worked one year at the Rio Xingu, which is one of the big tributaries to the Amazon, and it was wet season, and the river was kind of spread out, into the forest, and kind of this rushing river with these big boulders in the river, and there was a big pool by where our camp was, and the camp was actually set up by an engineering company. And then there was a big waterfall, and it was more of a rapids, kind of a sloping thing, and pretty dangerous waterfall. And we would get in a boat and go up the river every day, and then they'd let us off, and we'd spend time looking for things, and then they'd pick us up, and we'd come back down. And one time we came back down, and my wife wanted to get a picture of over the waterfall, kind of the forest from the top of the waterfall. And so the guy driving the boat headed towards the waterfall, and we got right above it, and the motor quit. And so all of a sudden we're drifting towards this waterfall. And, I think we all kind of turned white. And the guy driving the boat is back there trying to crank the engine. And so he's, desperately cranking the engine, trying to get it started again. And we keep drifting towards the waterfall, and we got really close to that rapids. And then he got the motor started, and he barely got us into the edge where the flow was slower, and we were able to actually make some progress up the river. But my comment was, no more waterfall photos.
Yeah, that sounds like a good advice.
Yeah. And we've had other experiences, occasional car wreck sorts of things. We had a student bitten by a Bothrops one night, and it got him with one fang through a boot. And so at about
in Central America or South America?
This was South America, so it was Bothrops atrox, real widespread Bothrops. Yep. And so we weren't sure whether we should take him to the hospital, which we knew was about 4 hours away on a muddy road. And it was raining, of course, and the road went up and down, this red clay kind of roads. And we finally decided that, well, we probably should, and not so much for the venom, because it was pretty clear he wasn't going to die. And but I started thinking about all the bacteria that are in some of these venoms, and so I thought, well, we better take him to the hospital. So we get in this pickup, and he's in the front, and he's got his leg up on the dash, and my wife and I are in the back of this pickup, and it has cloth on the sides, and it's raining like crazy. And so we're going on this road up and down hills, and rain coming in the back, and we're just totally soaked. We get to the hospital in this town, and there's a doctor there, and first thing she wants to do is give him antiserum. And so I tried to explain to her that he was allergic to everything in the book, she was not going to give him antiserum, and the bite wasn't bad enough to get antiserum, and that she should give him antibiotics, IV antibiotics. And so she went in the back room, and I guess she read some little scout book or something, and she came back and insisted on antiserum, and I finally had to stand between my student and the doctor and just say, it's not happening. And so, we ended up in a big argument. In the end, she didn't give him the antiserum. She did give him the antibiotics. She claimed he wasn't bitten by a venomous snake, and, with a room full of herpetologists. And so the next morning when we went in, there was another doctor that walked in, and this doctor was still there. And this other doctor took one look at his foot, and he says, "Oh, yeah, that's a fertilance bite." And he said, you did exactly the right thing. He said it wasn't a bad bite, and it wasn't, he didn't get any of the bleeding from the fingernails or any of that kind of stuff. He had a sore foot for about a week, and that was it. So
yeah, well, good. Glad that turned out well. So you were in Oklahoma for a huge chunk of your career, and now you're back in Arizona. I'm assuming you guys moved there when you retired. What brought you back to Arizona, and do you guys still kind of go on some of these adventures, or have you hung that up for now?
Well, we first retired to Florida, and to Citrus County in Florida, which is kind of rural. And we really liked that, but a couple things in Florida kind of got to us over time. And one was, the fact that there's so much water that everything is strip zoned. And so everywhere you go, it's stoplights and, stop and go traffic. So it's, kind of difficult to get anywhere. And so there was a little bit of that, and a little bit of the fact that it was just, the South, and it had the flavor of the South. And so we decided then to look in Arizona, and we looked around, kind of went to Tubac, and we liked Tubac, and so we came to Tubac. And when we retired, I sort of said, well, when we retire, I'm not doing any more science. I'm not going to write books. I'm not going to write papers. I'm not going to do analyses. I don't want to do any more of that. That ended up not being quite true, because I was collaborating with a lot of people, and I still do. But for the most part, I don't do most of the writing. I don't do most of the data analyses and so on. Mostly we use my data for different things. I had gotten in with Eric Pianca, who is an ecologist at University of Texas. And Eric and I, when we combined all the data we had collected, he worked mostly in Australia. We had a data set that was really, really good because we had, lots of different kinds of data for a lot of different species. And so when we put our data together, we were able to do a lot of stuff with them. So I continued collaborating. And what I do now, aside from collaborating, if you can believe this, I have a big mealworm colony. And my wife and I feed lizards in our yard. We have two parthenogenetic unisexual whiptail species that are common in our yard. And we have a spiny lizard, Scoloporus clarki, that lives in the mesquite trees. And we sit out in the mornings and hand feed mealworms to these lizards. And it's great watching them. They do all kinds of neat things. I don't know if you know about this pseudo-copulation behavior that some of the unisexual whiptails do, where they go through a behavior that looks like copulation. We see that all the time. We've actually seen that between species, which is strange. We chase roadrunners off when they get close. We have kind of a lot of fun with the lizard. We have a species of horned lizard in our yard. We're on 4 acres, and so we have quite a bit of property, and we're right up against national forest. So, we can hike out and be in national forest in 5 minutes. There are tortoise populations here. We see them on our property occasionally. We, of course, see Gila monsters. The first 3 years we were here, we ran drift fences with with minnow traps on the end. And we got 11 species of snakes and 11 species of lizards on our property.
Wow.
For a while, we were putting pit tags in the larger snakes. And we have one coach, but we kind of quit doing the fencing, I think 4 years ago or so. And some of the snakes are still around. We have one coach whip that's about close to 5 feet long. And it kind of hangs out under one of our cacti out in our property that has an old pack rat nest in it. So there's kind of a hole underneath it. And we see that snake every couple of weeks. We just saw him yesterday. And I caught him, I think, about 2 weeks ago and checked his pit tag. It's one of the snakes we put a pit tag in 2018, I think. And that snake has not grown 1 millimeter in all those years. And so that's as big as it's ever going to get. And so, we kind of entertain ourselves with that kind of stuff.
Yeah, well, it sounds like you guys are living the good life. So I hope it keeps going for a long time for you guys. And well, let's get to that snake story. So as I mentioned, I like to ask all of my guests to tell me a snake story. So what is your best snake story?
Well, I have a lot of snake stories, but I'll tell you one that was kind of a lot of fun. We were in an area by the Tapajós River in Brazil, and it was me, my wife, and 2 grad students, one a Brazilian and one an American. And we would go to this series of ponds every night that were in a stream bed that would dry up periodically, and it was usually dry, but there were these big pools, and we found things like Suriname toads in these pools, and we found there was a caiman that lived in one of them, a mata turtle, so kind of some of the really neat things that you can find, we found in these pools. And so we'd go out there just about every night and just see what we could see. And so one night we're out there, and there was a lot of leaf litter in the bottom of the pools. And there's a pool that was about the size of a large living room. And I was at one end of the pool, and the other three were at the other end. And I saw the tail of an anaconda sticking out of the leaf litter. And so my assumption was it was a juvenile anaconda. And so I grabbed a hold of the tail and picked it up, and the entire pond moved. And it turned out there was an anaconda in that pond that must have been somewhere between 16 and 20 feet long. And I, of course, yelled at the other three and told them to come down to where I was. And so they came down, and the snake just kind of started moving around in the pond, and the whole pond would move when the snake moved. And the head was probably 6 inches long. And the snake was as curious as we were. And so the snake would move around and it'd come over and it'd watch us, and we'd watch it. We spent 2 hours or so watching that crazy snake. And it went up under the bank at one point, and then its head came back out and looked at us, and then he came back out into the pond. And we spent half the night watching that crazy snake. And then the next morning we went out to see if the snake was still there. And the snake was gone, and so was the caiman. I think that snake probably ate everything big in that pond. But we've had a number of anaconda encounters, and that was one of the more fun ones.
Yeah, that wasn't it. What a exciting experience. So great. Well, I really enjoyed the conversation and chat with you today. I've learned a lot and heard a lot of great stories. So thank you again.
You're welcome.
And I just wanted to thank the audience and tell everybody to remember, snakes are animals too, and it's a privilege to see one in the wild.